Good evening and welcome. I'm Professor Rachelle Crosbie, and I have the honor of serving as chair of the Department of Integrative Biology and Physiology at UCLA. I'm also a professor of neurology in the David Geffen School of Medicine at UCLA. I wanted to mention a few housekeeping items before we begin. This event is being recorded. Please note that the chat function has been disabled. However, you can submit questions during the webinar using the Q&A window. Thanks to those of you who submitted questions in advance. We will get to as many questions as we possibly can. Today, I welcome you to our webinar, Looking for Memory Outside of the Brain, hosted by the Department of Integrative Biology and Physiology at UCLA. The department is focused on addressing the world's most pressing questions in physiology by harnessing our expertise and highly collaborative spirit. This evening, we'll hear from 3 speakers offering fresh perspectives on memory based on exciting discoveries from their research laboratories. It is my great pleasure to introduce the speakers who will be presenting this evening. I'm pleased to introduce to you our first speaker, Dr. David Glanzman. Dr. Glanzman is a distinguished professor of the Department of Integrative Biology and Physiology and a professor of neurobiology at UCLA. The Glanzman Lab is interested in cell biology of learning and memory in simple organisms. Specifically, the Glanzman Lab uses the marine snail and the zebrafish as model systems. Through the study of simple forms of learning, such as habituation, sensitization, and classic conditioning, Dr. Glanzman hopes to gain insights into the underlying biology of learning and memory in more complex organisms, including humans. The Glanzman Lab is also interested in how memories are maintained in our brains for long periods of time. Dr. Glanzman earned his BA in psychology from Indiana University Bloomington and followed by a Ph.D. in psychology from Stanford. Please join me in welcoming Dr. Glanzman.
Thank you, Rachelle. Let me share the screen. This webinar is sort of advance notice of a symposium that we are going to have in June. And the title of the symposium is Pushing the Boundaries. And it's being funded by a generous grant from the Luskin Foundation and also by funds from the UCLA Life Sciences. Now, some of the speakers that are going to be at the symposium are shown here. And these speakers include philosophers, they include systems biologists, they include biomedical engineers. And the purpose of this meeting is to, as the title suggests, is to present new and challenging ideas in biology, neuroscience, and artificial intelligence. So some of the topics that will be addressed at the meeting are synthetic life, cognition in plants, inheritance of memory, which we're going to hear more about this evening from Dr. Murphy. And so the purpose of this meeting will be to present ideas from a variety of related fields that challenge current thinking. And some of you may have heard that people have argued there is a decline in disruptive ideas in science, a relative decline. And disruptive ideas are ideas that are outside the box, that tend to overthrow accepted paradigms in science and lead to new paradigms. And People argue about the reasons for this, but my hope in putting together this meeting was that that we could further the propagation of disrupted ideas in these fields. So most of us believe that, or it's widely believed that memory is encoded in the brain as changes in the strength of the connections among neurons, that is, the synapses. And and the idea is that as an organism learns, or as a person learns, that there are alterations in synaptic connections, and learning experience reshapes the synaptic connections, and then this reshaping of the connectivity of the brain is how the memory is encoded. However, since about 1960, there has been a competing idea, which is less popular, which is the possibility that memory could be encoded as changes in molecules. And one of the first proponents of this idea was a psychologist by the name of James McConnell. And he worked on an unusual organism for a psychologist. And what he worked on was the flatworm planaria. And this worm has a nervous system which comprises a brain and ventral nerve cords. And McConnell, in a pioneering experiment, showed that you could bisect this animal, and the head would regrow a new tail, but more surprisingly, the tail would regrow a new head and a new brain. And McConnell trained a group of these planarians on a task known as a classical conditioning task. He paired a light with an electric shock, and after several pairings of the electric shock, whenever the light with the electric shock, whenever he gave the light, the worms would contract. And then he bisected the worms and allowed the tails to regenerate, as shown in this slide, to regenerate a new head. And what he found was that the regenerated worms were exhibited memory savings. They appeared to preserve the memory of their training, even though they had to regenerate a brand new brain. And the second speaker tonight, Dr. Mike Levin, has repeated this experiment of McConnell's and found the same thing. Now, McConnell suggested that what his results meant is that memory could be stored as molecules, as changes in specific molecules, and those molecules might be distributed throughout the worm's body. And he suggested that the molecule that is involved in conserving the memory might be RNA. And this idea flourished for a while, but by the end of the 1970s, it died out. And the synaptic model has been the synaptic model of memory has been the one that's persisted. However, recently, there's been more interest in this idea that memory could be encoded as changes in molecules. And my laboratory has recently published an experiment that used this organism shown here. It's a marine snail. And what we did was give these snails a series of electrical shocks to their mild electrical shocks to their tails. And this produced an enhancement of their reflexes in response to touch. So this enhancement is known as sensitization. And after training the snails, we extracted RNA from the trained snails and injected it into snails that had not been trained, that had not received electrical shocks. And what we found was that the recipient snails showed evidence of memory. They showed sensitization. By contrast, when we took the RNA out of untrained donor snails and injected that RNA into untrained recipients, the animals, the recipients did not show sensitization. And in additional experiments, we found that a type of so-called epigenetic change appeared to be critical for this memory transfer. And in particular, the epigenetic change that was involved was DNA methylation. So the question is now, what are the potential molecules that might be involved in this memory transfer? So with that, I'm going to turn it over to my next speaker, but you're going to hear more about this planarian in the next talk, and you're going to hear more about RNA and memory, and in particular, the transfer of memory in the talk by Dr. Murphy, which comes next.
Thank you, Dr. Glanzman. I'm excited to learn more about disruptive science. Next, I have the pleasure of introducing Dr. Mike Levin. Dr. Levin is a distinguished professor and holds the Vannevar Bush Chair in Biology Department, and he serves as director of the Allen Discovery Center at Tufts University. He's also an associate faculty member of the Wyss Institute at Harvard. Dr. Levin's work focuses on the scaling of intelligence in unconventional substrates, from single cells and slime molds to tissues, organs, and whole organisms. Specifically, his group studies how memory, decision-making, and creative problem-solving are implemented in a range of natural, artificial, and hybrid systems. Using a combination of developmental biophysics, computer science, and cognitive science, Dr. Levin has transitioned his conceptual frameworks and computational models to novel regenerative medicine applications targeting birth defects, injury, and cancer. His laboratory also creates and studies new synthetic life forms to understand diverse intelligence in novel embodiments. Dr. Levin earned his BS in computer science and biology from Tufts University, followed by a Ph.D. in genetics from Harvard Medical School. Please join me in welcoming Dr. Levin.
Thank you so much. Very happy to be here and share some thoughts with you on some unconventional selves and the kinds of bioelectric memories that they can hold. One of the remarkable facts about developmental biology is that all of us made the journey across the so-called Cartesian cut. We were all at one point just physics, so a quiescent oocyte, something like this. And then through this incredible process of embryonic development, we become one of these organisms, or perhaps even a human with high-level intelligence, metacognition, and all of those kinds of things. So it really, to understand what we are, we really must be able to follow this process backwards, because it's a very slow and gradual process, and ask ourselves, what is it that happens during this that takes us from physics to cognition? And what's interesting is that, of course, all intelligences are collective intelligences. Rene Descartes was very interested in the pineal gland as kind of the center of what makes humans unique, because it was he said that it was the one structure in the brain that is not duplicated in terms of left and right. It's a single structure. But if he had access to microscopy, he would have looked inside this pineal gland and found out that it's made of cells. And inside of each one of those cells is all of this stuff. So everything is made up of parts, and the kind of remarkable tasks before us is to understand how these parts add up to the intelligence of these cells, which in turn sums together to form this emergent intelligence of humans and other animals. We are all made of single cells, and this is a single cell here. This is a free living organism known as a lacrimaria. And you can see here that how remarkably competent this little creature is in its own local goals. So no brain, no nervous system, but all of its anatomical, physiological, metabolic needs are being handled at the single cell level. So what's amazing about biology is that we are not merely structurally nested dolls of molecules and cells and tissues and organs and all the way up to collectives of swarms. But in fact, each of these layers solves problems in its own space. So there are spaces of anatomical structures, spaces of gene expression or transcriptional space, spaces of possible physiological parameters, and of course, the three-dimensional space of classical behavior. And biology uses systems at each one of those spaces to solve various kinds of problems. Now, when I talk about these different types of systems being intelligent, what I mean is the definition of intelligence by William James, which is the ability to reach the same goal by different means. And of course, there are different degrees of competency in those. So I want to show you, take a couple minutes and show you some specific kinds of competencies of one of my favorite unconventional intelligences here, and that there are many, which is in particular this, the morphogenetic agent, the collective intelligence of cells that builds and repairs individual organisms. Here's one simple example. As we start thinking about intelligence as the ability to reach the same goal by different means, we can ask ourselves, how does this work under different challenging circumstances? So we know that embryonic development is incredibly reliable. Most of the time, acorns make oak trees and frog eggs make frogs. But so we can challenge this process and ask what kind of capabilities it really has. So this here, what you're seeing is a cross-section of a tubule, a little tube that goes to the kidneys of newts, of salamanders. And normally there's about 8 to 10 cells that make up this tubule. One thing we can do is artificially increase the amount of DNA in these cells. The first remarkable fact is that if you simply increase the amount of genetic material, nothing much happens. You still get a normal newt out of it, but the cells get bigger. So they adjust their size based on how much DNA they have. And when they adjust their size, the same number of cells don't fit in the tubule anymore. So it's automatically fewer cells will participate in this process to make the exact same tubule. So that's the second remarkable thing, is that the cells will actually scale their number to the problem at hand. The most amazing thing happens when you make these cells so enormous, and this is 6 to 8 times the normal DNA that these cells are supposed to have. When you make them so enormous that one single cell will bend around itself to form the same lumen. So what you see is, in the service of this large-scale anatomical goal of having a particular tubule, different molecular mechanisms get called up to execute this activity. And this is very important. It shows you that this process of morphogenesis, of building shape, can adapt to novel circumstances. The system can't rely on having the right amount of DNA. It can't rely on having the right size of cells, but it has to be able to produce the same outcome every single time. There's another system that does that. We discovered this a few years ago, where these tadpoles, they have to become frogs. And during the normal process of metamorphosis, they rearrange their face. So the eyes move forward, the jaws move, various things move. Well, it turns out that this is not a hardwired process whereby every organ just moves where it's going to go, the same distance in the correct path. We made these so-called Picasso tadpoles, where all the organs are in the wrong place. So the mouth might be off to the side, the eye might be on the top of the head, everything is scrambled. And these animals still make perfectly good frogs, because all the different organs will move through novel paths to get to the correct shape, and then they stop. So what the genetics specifies is not a hardwired system. It specifies actually an error minimization scheme. It has a certain amount of intelligence to get to the same final goal from different starting positions. And so this raises an important question that we'll talk about momentarily of how does it know when it's right? And so finally, during adulthood, some animals, such as this axolotl, are able to regenerate and repair many body organs. And so what happens in the limb is that if the limb is lost at any position along this axis, the cells will very rapidly regrow exactly what's needed. So this collective intelligence of these cells navigates that anatomical morphospace until it gets to the correct configuration, and then it stops. Okay? That's kind of the most amazing thing about regeneration, is that it knows when to stop. So for some years now, we've been asking this question: how does it know what the correct pattern is? Right? What allows this collective of cells, which are now working on this large construction project, to know where they're supposed to go? And it turns out that very much like in the brain, and so this is electrical signaling in the zebrafish brain that carries all the intelligence and the behavioral competencies of this animal. This is all run by electrical machinery in these neurons. Well, it turns out that this electrical machinery is ubiquitous. It's present throughout the body. These ion channels, these electrical synapses are everywhere. Evolution discovered this long before nerves and brains came on the scene. And what we can do is we can, much like neuroscientists are trying to do neural decoding in brains, we can do neural decoding in embryos, the rest of the body tissues, to understand what this collective intelligence is doing in its building projects. And so one of the things we can do is we can read now these memories of the body, and we can see this, we call this the electric face. This is a voltage sensitive dye, which is literally telling us where all the cells of the face are going to build various organs, eye, mouth, and so on. And so if we reproduce artificially using optogenetics and other techniques, we reproduce this pattern somewhere else. It's very easy to convince, for example, these gut cells that they need to make an eye. And this eye will have all the right internal components, even though we never have to specify them. All we have to do is say, build an eye here. And in fact, it has this remarkable capacity of recruitment, like ant and termite colonies, where if we just inject a few of these cells, they will recruit their neighbors to make this very nice lens of the correct size. Because these cells together know there's not enough of them to make a proper lens, they recruit their neighbors. The blue ones are the ones that we manipulated. These are perfectly normal cells that get recruited. And so now I'll just close by telling you two very quick stories. So this is these are the planaria that you just heard about from Professor Glanzman. And the amazing thing about planaria, of course, is that you can cut them into pieces. Every piece gives rise to a correct little worm. And so we asked this question of how does a middle fragment, so let's say you cut off the head and the tail, and here's this middle fragment, how does it know how many heads it's supposed to have? And it turns out that there's an electrical circuit that we can visualize. So here, this is the circuit that says build one head, one tail. And we can go ahead and rewrite that pattern. And we can say, actually, a correct planarian should have two heads. And we can do this in the one-headed body. And when you do this within the one-headed body, nothing much happens because it's a counterfactual memory. It's not a memory of what's happening now. Until you do that amputation, at which point it goes ahead and builds what the memory says. It builds the two-headed worm. So a single one-headed body can have two different representations of what a correct worm should look like. So we're literally getting a glimpse inside the memory of the collective intelligence of these cells at the anatomical level, as we can read what it thinks a correct body looks like. And I call it a memory because when you amputate these two-headed animals, so you get rid of the primary head, you get rid of this ectopic secondary head, even though their genetics are untouched, the hardware is exactly the same as it was, that middle fragment will continue to give rise to two-headed animals forever. And so you can see here these two-headed animals. So the question of how does a tissue know how many heads this animal is supposed to have is not set in the DNA. It's actually, it can be rewired by experience, not by manipulating the hardware. And so I'll just end here by saying that, by showing you, first of all, some xenobots. So these are autonomous little organisms formed from frog skin cells that, given some freedom from the rest of the animal, they're able to re-envision their kind of multicellularity to the point where they have new behaviors like collecting loose cells into the next generation of xenobots. They have this bizarre form of replicating themselves. And just to say that there's this incredible option space of new bodies and new minds, hybrids and cyborgs and all kinds of combinations of evolved material, design material, and software, where all of the known natural biological beings are a tiny corner of the space. And the future of this field is going to be to use this massive massive field of novel living beings to understand different ways that intelligence can be embodied in this world. Thank you so much.
Thank you, Dr. Levin. I love the concept that we all started off as just physics in the single cell state. I'm now pleased to introduce our last speaker, Dr. Coleen Murphy. Dr. Murphy is the James A. Elkins Jr. Professor in the Life Sciences. Dr. Murphy is also a professor of molecular biology and the Lewis-Sigler Institute for Integrative Genomics. Additionally, she is the director of the Paul F. Glenn Laboratories for Aging Research at Princeton University and director of the Simons Foundation's collaboration on plasticity and the aging brain. The Murphy Lab focuses on the process of aging, which remains a fundamental mystery of biology. The goal of the Murphy Lab is to understand the molecular mechanisms governing longevity and the maintenance of the biological processes that exhibit age-related decline. Dr. Murphy earned her BS in biochemical and biophysical sciences from the University of Houston, followed by a Ph.D. in biochemistry from Stanford University. Please join me in welcoming Dr. Coleen Murphy.
Thank you for having me. I'm sorry, here we go. No. Sorry, something's gone wrong. All right, so today I'm not going to talk about aging, but in fact, a different phenomenon that we discovered while we were looking at our favorite organism, which is C. elegans. So the main thing you need to know for this talk is just that C. elegans has a rapid generation time. So as David alluded to, we are interested in looking at what happens across many generations. And since these animals can go from an egg to an adult in less than 3 days, it allows us to look at many generations very quickly. They're also a very simple organism, they have a very simple body plan. But what's really cool about them is that about 75% of their genes and proteins actually are conserved with humans. On top of that, they have a rich set of behaviors, including learning and memory, which we have studied in other ways in our lab. But in the lab, the worms eat E. coli, but out in the real world, worms are eating bacteria that they find on mostly rotting fruit and in compost piles. And in fact, they almost never meet E. coli, which we feed them in the lab, but in fact, they have a really broad variety of other bacterial species that they encounter. And about a third of those are Pseudomonas species, which I'm going to talk about. And the reason those are interesting is because they are, they can be either beneficial, like a food source, in fact, it's a major food source, or detrimental. When I say detrimental, what I mean is that if we feed them this clinical isolate of Pseudomonas aeruginosa called PA14 for only 24 hours, you can see that they're dying really quickly. Over here on the right, they're really sick. And in fact, they're only going to live for 2 to 3 days more. While on the left, this is what they should look like, nice and healthy. All right, so the weird thing about worms is that they really love the smell of Pseudomonas. And so when they're confronted with Pseudomonas, they actually will all choose that direction. So that's this choice arrow down, means they choose Pseudomonas, despite the fact that it's really bad for them. But if you train them for a little while by feeding them on the Pseudomonas for a couple of hours, then they learn to avoid it. So this is actually a learned avoidance. All right. So what my graduate student, Rebecca Moore, wanted to ask was, what happens to the progeny of the mothers that have learned to avoid? And so she looked at those progeny by taking their eggs and then developing them on E. coli and asking them, even though they've never seen Pseudomonas before, what do they do? And what we found was that even those progeny they already know to avoid them. So this is really strange. This is intergenerational avoidance. Okay. So we asked how many less generations does this behavior last? And we see that the grand progeny, the great-grand progeny, the great-great-grand progeny all already know to avoid Pseudomonas, even though they've never met them. And it's only in the F5 generation that they go back to their innate and the naive attraction to Pseudomonas. Right, so that's shown here. This is how many generations they avoid. And so in the wild, this might be akin to, being out in the apple orchard for 2 weeks when there's a heat wave. So they might want to not hardwire this avoidance of a very good food source, because it could go back to being a good food source once the temperature drops. So we wondered what the animals are reading from the bacteria. And just to cut a long story short, we figured out that if we took DNA and RNA out of the bacteria and just fed it to worms on E. coli, we could actually mimic the effect. That's what's shown here on the right. The total RNA was able to give the same effect as training them on Pseudomonas. And in fact, if we broke that RNA into large and small RNA, we found that the small RNA was sufficient to induce all the training. It caused all the effects that we showed, except it never makes the worm sick. So just being fed this RNA was sufficient to give them this effect. And actually, it induces the 4 generations of avoidance as well. So to try to figure out what's in that mix of small RNAs, we just did some differential expression analysis. That means we compared what's going on at the small RNA level under different conditions. And we only found 6 small RNAs that are different in the condition that causes this avoidance behavior. And so we were able to test each one of those, and that's shown here. And only one of them was sufficient to induce all this avoidance behavior. And it turns on, it affects the brain in the exact same way as we'd seen before with the bacteria. And it induces all 4 generations of this avoidance behavior as well. So what is that small RNA? Well, when we looked at it more carefully, what we discovered was the small RNA from the bacteria actually has a perfect match to a gene in C. elegans. And that gene in C. elegans is known to have effects in the brain. So it's required for things like neuronal transduction excitability and presynaptic transmission. And we see that it actually is downregulated when the worms meet that Pseudomonas, the clinical isolate. All right, so what we think is going on is this: worms eat bacteria and they take out the small RNAs, including this P11 RNA, that the bacteria are making for their own purposes. And then they basically transmit this signal, they translate it and turn it into a different kind of signal that goes through the entire worm from the intestine to the germline. And that transmits the signal to the neurons. And then when it does that, there's a switch that basically tells the worms to go from attraction to avoidance, right? And what's cool about that is that it does this by basically the worms are using this P11. They think it's a biomarker of future infection and pathogenesis. So they know that the worms are going to get sick. And so they should pass on this information. All right, and so in this last 2 minutes, I want to tell you about the part that David was referring to. We were wondering if you could actually transfer memories from the animals that have learned something to naive animals. And so we actually took the grand progeny of the trained mothers and we ground them up and we squirted that onto naive animals. And we found that that was sufficient to induce this learned avoidance, right? So there's something in the lysate of those grand progeny. So we wondered what carries the memory. We did some very, very simple biochemistry and just took the heaviest fraction, and we can see that that fraction was sufficient to induce the memory as well as the 4 generations of avoidance. And so we just asked, well, what's in that fraction? And this is, these are EM images, and we see these little viral-like particles, the little capsids in that EM. And so we wondered what could be causing that. And what we discovered was there's a retrotransposon called SIR1, which seems to make these little viral-like particles. And if you don't have those SIR1 particles, you don't get any of the effects that we saw from the lysate or the isolated viral-like particles. So we really need that in order to function. So what we think is going on is that these viral-like particles are carrying some information from these processed small RNAs from the germline of the worm to their neurons, and that's carrying information to them. So finally, we wanted to ask the craziest thing, which is, can they actually share this information with one another without being ground up? And so to do that experiment, we took those same grand progeny animals, but instead of taking the worms, we just take the media that they've been swimming in. We apply that to the animals. And again, that was sufficient to induce transgenerational avoidance of these pathogens. And again, it required the SIR1 retrotransposon in order to do that. So we think what's going on in the wild possibly is that these capsids can carry this information to neurons, but at the same time, if the worms are super sick and dying and exploding, that means that other animals in their neighborhood could take up that information and learn from, this other mother's mistake and essentially learn to avoid and carry that information on for several generations as well. And so I just want to thank the people who did the work, Rachel Koletsky in my lab and Rebecca Moore. They worked really hard to do basically that was a set of 3 different papers. with help from other people. And of course, we got generous funding from lots of institutional grants, and we had others who shared reagents with us. And with that, I will stop sharing, and so we can move on to the next part. Thanks.
Thank you so much, Dr. Murphy. Such elegant data for the concept of generational memory. I'm really blown away by that. So now I'd like to invite Dr. Glanzman and Dr. Levin to join us again and open up for questions from our guests. We will try to address as many of your questions as possible. Please click on the Q&A window at the bottom of your screen and submit your questions in the Q&A box. And just to get started, we'll go through some of the questions that were submitted in advance of the webinar. First one is, could the panelists or the speakers comment on memory in the context of trauma like PTSD and Alzheimer's?
Okay, well, with respect to PTSD, you can think of PTSD as a sort of turbocharged memory. And the mechanisms that underlie PTSD are in many of the same biological mechanisms that underlie standard learning and memory. But however, these memories are, the PTSD memories are exceedingly persistent. And this raised the question of why are they so persistent? And investigators are trying to get a handle on that. And the other question of clinical interest is there a way to ameliorate or even perhaps eliminate the memories that give rise to trauma? And various approaches are being taken to try to do that. And while they've had some success in the laboratory, I think it's fair to say that clinically, it's been very difficult to eliminate these memories entirely. With respect to Alzheimer's, it's somewhat of the opposite problem. It's memories that gradually disappear. And for people who, you are familiar with anyone who's had Alzheimer's, you know how distressing it is, both for the person, him or herself, and also for the family members. It's an extremely devastating disease, and we know it's due to physical deterioration of the brain. And people are attempting to block molecules, amyloid beta, aberrant molecules that seem to underlie the brain deterioration. And again, this is a disorder that so far people have not succeeded in dramatically ameliorating. And I think what this both PTSD and Alzheimer's show is that we still have a lot to learn about the basic biology, the cell biology of memory. And I think some of the work that you've heard tonight will eventually contribute to potentially the solutions to these problems. These are very difficult and distressing problems.
Thank you for that, Dr. Glanzman. Another question from our audience is that given that memory may be expressed in single cells, and we saw many beautiful examples of that here today, are there any ways or thoughts for how to improve memory and prevent memory decline?
Well, one possibility I think is quite fascinating is that the idea that memory is encoded in molecules and may be stored within cells rather than at synapses has certain implications. And with respect to Alzheimer's, one of the things that happens in the early stages of Alzheimer's disease is that these aberrant molecules, amyloid beta and others, cause the destruction of synaptic connections. And this destruction of synaptic connections correlates with the gravity of the memory loss. But what if the memory isn't stored in the synaptic connections, but it's actually stored in the neurons themselves? If that were the case, then it might be possible to regenerate the appropriate synaptic connections among neurons and restore memory. In the laboratory, people have succeeded in reactivating memories that by all behavioral signs had been completely erased. And so this may lead to treatments for at least the earlier stages of Alzheimer's. Of course, in the later stages, there's actual death of neurons. And after that, the cell's not there, you're not going to have memory.
Thank you for that. This next question is for Dr. Levin. And that is, can you explain a little bit, two questions. Can you explain a little bit more about the cutting of the worms? How is that achieved? And can you clarify they actually regrow completely back to normal? That's the first question. And the second question is a bigger question, and that is, what would finding memory outside of the brain mean for the field of science and health? That's a big one.
Yeah. So let's see. The first thing about the planaria is that many species of planaria reproduce by ripping themselves in half. So cutting the planaria, which we normally do for convenience with a scalpel, so you just come along and you cut them. The planaria have no issues with this because this is how they normally reproduce. So what normally will happen is that the tail end will sort of stick down to the dish, the head will take off, and they rip themselves in half, and then they regenerate, and now one worm becomes two worms. And it's a remarkable fact that you can cut them into many pieces. The record, I believe, is something like 276 pieces, and every piece reliably gives rise to a perfect little worm. In addition to that, they appear to be immortal, so there's not any evidence of aging at the level of the organism. They're extremely cancer resistant, and there are many interesting aspects of its electrophysiology that I think underlie that. But what it would mean for medicine is this: if it is true that intelligence of a kind that it can learn from experience, can take specific actions in various problem spaces, exists outside the brain in other tissues, in molecular networks, and so on. This opens up a whole new opportunity for medical intervention, because right now, molecular medicine is all about the hardware. So people are very interested in editing the genome, protein engineering, rewiring pathways. This is all at the hardware level. And if there's one thing we've learned from the journey of information technology in computer science from the 1940s and 50s, where you had to program a computer literally by physically rewiring it, what we've learned is that for many of these systems, there's an incredibly powerful path which is reached by taking advantage of the device's reprogrammability, the software level. And I think that there are many ways that we can think of to intervene in various diseases top down by literally communicating with the cellular collective to cause it to move towards regions of physiological space, of transcriptional space, of anatomical space in the case of regeneration, that we will be able to achieve that top down by respecifying the targets for these cells much easier than we would ever be able to do by micromanaging it bottom up.
Thank you for that. Another question, this is a great question from someone in the audience, and that is, I'm interested in hearing the panelists' insights about the transfer of memory and evolution. It certainly must represent an advantage to inherit our ancestors' memories, and there must be some sort of positive selection enhancing these mechanisms. I'd like to open it up for a discussion with the panelists.
I'd love to answer that question. So I think we can see in nature there's some advantage. For example, in the case that I talked about today, which we found is true for both using wild C. elegans and wild bacteria. So we know that this is something that must happen in the wild. But I think I would caution everyone about interpreting this too far, because in fact, I think there's an important concept about the scales of generations and the time of memory and the environment. So in a worm's world, right, the time of the generation is so fast that it's possible that multiple generations will be exposed to the same sort of environmental conditions that they need to learn about. But once you get up into long-lived organisms, this sort of becomes a little bit unmatched, right? So if there is transgenerational inheritance of learned behaviors or information in mammals, especially humans, I think it all has to be maladaptive, sort of an epigenetic hangover from things that we had as very simple organisms where it was useful. And now we still have many of the same molecules and same regulators, but it's not necessarily useful to us. And that's why it's important to learn about the mechanisms so that we can break them in cases where it's not useful to us. We want to get rid of it. So we want to understand it a lot better.
That's really great.
I'd just like to,
no, please.
I'd just like to follow up on that. There's evidence that there is transgenerational inheritance of memory in mice, at least. And Isabel Monsoy from Zurich is going to be one of the speakers at the Pushing the Boundaries Symposium, and she's going to talk about her research that suggests that there's inheritance of acquired experience in a mammal, the mouse.
Thank you, Dr. Glanzman. Another question from our audience, and this is for Dr. Murphy, and that is, could the mitochondria from the C. elegans be the unit that carries the information about trauma danger that's passed down from generation to generation? Do you think there's a role for the mitochondria?
That's a really interesting question that we haven't asked in my lab, so I'm not going to say that we know one way or the other. And I don't want to discount it, because every time I feel like I need discounting mitochondria, it turns out it crops up in some other way. It is true that mitochondria in C. elegans, about something like over 90% of the mitochondria in C. elegans are in the germline. And so you can imagine that if something impinges on the germline of the oocytes of the animal, it could be passed on to the next generation. So I imagine that the way that something like that could happen is if you have something where there's an interaction between the small RNAs that we're studying and the mitochondria themselves. And so we don't have any evidence for or against, but that's just because we haven't tested it. So it's an interesting question.
I love it. And I agree with you that the mitochondria seems to be involved with everything, never discounted. Another question from our panelists is about pain. Is it possible that the memory of pain can be stored and processed outside of the nervous system?
Coleen, you want to try that? Or
I have no insight into the pain at all.
okay.
So
yeah, I mean, it's stored outside the nervous system, since pain, there's a literature on pain suggesting that pain really is indeed a type of memory. And many of the same mechanisms that are involved in memory, for example, the memory that I talked about in the snail sensitization, those same mechanisms are also involved in pain. And the molecular biology of pain is very similar to the molecular biology of certain types of memory. And consequently, if one thinks that memories can be present outside of the nervous system, then I see no reason why pain, the molecules that are involved in pain wouldn't also, at least potentially, be outside of the nervous system. Having said that, I'm not aware of any findings that suggest that.
Thank you, Dr. Glanzman. It was mentioned in some of the talks about single cell RNA sequencing, and that's in the press a lot, and also these new technologies like spatial transcriptomics. And given that that there's this beautiful data on the study of memory and its link to RNA, can the panelists comment on these state-of-the-art technologies? What are they, and how can they help us investigate memory outside of the brain? .
Well, a lot of these technologies are quite amazing in allowing us to detect many more aspects of the hardware of the system. So the various RNAs, the proteins, metabolites, lipids, all kinds of things that are present there. That's so, and they're opening all kinds of new inroads in understanding some of the information molecules that exist. One thing that we really need a lot more of, from my opinion, is similar technology to profile a lot of the physiology of these cells. So physiomics. So basically understanding the bioelectric states, the other types of physiological kinds of states that are actually not detectable in the dead state. So when your cell is dead, you can sequence the DNA, you can look at the proteins, you can look at the RNAs, you can look at all of that. There's a ton of stuff, including for example, the bioelectric states that disappear completely the moment the cell dies. And so this is only doable in the living state, and physiomics really has lagged far behind some of these other biochemical technologies. And I sort of really, we work on this, and other people are working on this too, to really pull it to the same level.
Love that. I love that. This next question from the audience is directed to Dr. Glanzman, and that is, maybe the RNA that's transferred from the Glanzman snail, I like that it's called the Glanzman snail, maybe the RNA is related to the bioelectrical circuitry changes in the receiving organism. For instance, does it encode an ion channel or something like that? So can you tell us more, Dr. Glanzman, about what is the RNA doing? What is it encoding? Can you drill down on some details?
Yes, I wish I could. So far, in the experiments that we did, so far we've just injected whole RNA into the snails, and I've recently gotten some grants from NSF that will enable us to attempt to isolate the so-called mnemonic RNAs, the RNAs that we think are involved. We believe, however, that the RNAs are involved are so-called small RNAs. These are distinct from the messenger RNAs or the coding RNAs that are involved in genetic transmission. So we haven't gotten even to the point that Coleen has gotten with her work in identifying some of the critical RNAs. That's something that we hope to do in the future. So I'm sorry to disappoint the questioner, but science depends on money, and if you don't have money, you can't do science. I mean, that's just the way it is.
Thank you, Dr. Glanzman. Another question for the group, and I love this question. Are the molecular markers and the RNA that's been identified through the 3 different talks that we've heard, are these sexually dimorphic?
The behavior that we look at is kind of sexually dimorphic, because the males of C. elegans, which need a lot less food because they're not making progeny, they already avoid all these pathogens. So they don't need to switch. But what we found kind of surprising to us, we thought that when the animals mated, it would wipe their memories clean, and it doesn't. It actually passes on, and you can train males on the Pseudomonas and their progeny inherit. So we're currently trying to understand that better, because it's entirely possible that's in completely different mechanisms from what I told you about today. And so we're looking at that in more detail, but we were surprised that they actually could do it. So there's some sexual dimorphism there, but maybe the question, there's one. We're interested in a different aspect of the question.
Very interesting. Okay, we have one more question, and I'd love to hear from all the panelists, and then I'll close with a few remarks. And that is, where do each of you see the future avenues of investigation to explore memory outside of the brain? Dr. Murphy, would you like to go first?
Well, for our work, there's a lot of obvious directions to go in, and some of the questions here, and like some people have asked the really great questions about like how often does this happen in nature and how many different ways could it happen. We would love to see whether the same mechanism happens in, for example, Drosophila and higher organisms as you move up the evolutionary scale. how well is this kind of mechanism conserved? And in fact, we're kind of interested in whether the bacteria in our gut, whether our cells actually take in RNA from those bacteria, because it could be affecting us in different ways. So that's more the transkingdom signaling, but it could be using some of the same mechanisms. So that's where we're going with this work.
Dr. Levin, we'd love to hear your thoughts.
For me, I'm really interested in how the different kinds of memories and these different kinds of agents relate to each other. So, for example, we've been studying memory in the morphogenetic agent in terms of bioelectrical pattern memories. We've studied some behavioral memories in slime mold and in planaria, as Dr. Glanzman said. We've also found memory in gene regulatory networks. So basically, collections of genes that turn each other on and off can also learn. They have 6 different kinds of memory. And so every layer of this thing seems to have its own type of memory. And so to me, the most exciting thing going forward is to figure out how all these different memories relate to each other.
For me, I've spent most of my career believing in the synaptic model of memory, and so it was surprising to me to come up with the evidence that maybe memory isn't stored as changes in synaptic connections, even though there's no question that when an animal learns something, there are changes in the synaptic connections. But the idea that this isn't how memory is stored, that perhaps memory is stored as a molecule, has all sorts of profound implications for thinking about how you can cure memory-related disorders, and how, for example, a fascinating question is, if memory can exist outside the brain, how long can it exist for? I mean, I think that's an incredibly fascinating question as well. And some of these molecules are reasonably long-lived, so that's a fascinating question. But it's important to understand, because we know that synapses are changed during memory, and we know that synaptic change is important for memory. If memory is encoded, for example, in the nucleus of neurons as molecular changes, how does the nucleus sculpt the synaptic change? Is that a possibility? And if that is a possibility, how does the nucleus know which synapses to change? I mean, for me, I think that's a really fascinating question that has to be addressed if you're arguing that memory is encoded as these molecular changes. How does that interact with the synaptic changes? Is that the driver of the synaptic changes? And if so, how's that even possible? I mean, how would the nucleus know which synapses have to be strengthened, which have to be weakened? Is there some bidirectional communication from the synapse back to the nucleus and vice versa? So for me, those are interesting questions.
Thank you. That's really great. I'd like to thank everyone for joining us today. We hope you enjoyed these disruptive presentations and learned something that you will take with you. As a reminder, this webinar has been recorded. We will send a post-event email to everyone who RSVP'd with a link to the webinar recording. Please feel free to share it with others. The webinar will also be posted on our departmental website. And we hope that you will join us to learn more about these exciting disruptive topics at the Luskin Endowment for Thought Leadership Symposium on our UCLA campus in June. There will also be opportunities for remote attendance. A link with additional information about the symposium will be shared in the post-event email. I'd like to give our presenters a huge round of thanks and applause. And to you, thank you for joining us tonight. Hope everyone has a wonderful evening. Bye.
Thanks very much. And thank you to the audience.
Thank you, everybody. Thank you.
Yes, thank you. Bye.